afternoon, everybody, and thanks for joining us for another in our series of Live from the Lab at La Jolla Institute. I'm really pleased to join you today, and I want to thank you all for joining us. I'm Eric Zwisler. I'm the board chair at the La Jolla Institute, and hosting this this event. I am unarguably the least important person that you're going to hear from today. And with that, it's my pleasure to host this tremendous group of people who've been working quite hard over the past year and exploring the science behind COVID and viral infectious diseases. We'll get to that in a minute. Right now, just if we could have the next slide, just to lay out where we're going today. We're going to hear first from Dr. Mitch Kronenberg, who's our president and chief scientific officer. Mitch has been with the Institute and led the Institute for over 15 years and is the heart and soul of our Institute. And then Steve Wilson, our esteemed chief operating officer and executive vice president, is going to help us summarize some of the main learnings and insights that we've developed and others have developed to put the COVID pandemic in perspective. After that, we'll take a look at part of our labs involved in infectious disease areas with Alba Grifoni, and follow up with questions and answers. And just a reminder to everybody, there is a chat function or a Q&A function on the bottom of your screens. I see we already have people typing in questions. We really look forward to your questions, and hopefully we'll have time to answer more of them. I'll be moderating that session of the discussion. Next slide, please. It has indeed been the best of years and the worst of years. Unfortunately for the Institute, the pandemic hit a very good portion for us, or good part of our research in our infectious disease and vaccine research area. And so we found ourselves quite well plugged in with a lot of experience already in not only immune response, T cell and antibody responses, but also specifically with coronaviruses. And then to top it off, also with quite an experience in vaccine development and the ins and outs of vaccines, efficacies and design. A lot of that work has been recognized in many of the scientific and mainline publications that you see on the screen before you. And indeed, this has been essential in raising the visibility of the La Jolla Institute, unfortunately around a pandemic, but as I said, an area in which we've been able to be a small part of the international effort to develop vaccines and to confront this pandemic. So for that, we're happy, and for that, we're happy that we recognize you and know you and your interest in the La Jolla Institute and what we're doing. With that, I'd like to get on with the main body of our presentation and introduce to you Dr. Mitch Kronenberg. Mitch is our president and chief scientific officer, and the La Jolla Institute is what it is today because of Mitch's investment, his vision, and his leadership of the organization. So without further ado, I'd like to hand it over to Mitch to lead us through the scientific presentation. Mitch.
Thank you. Thank you very much, Eric. And good afternoon, everyone. Thank you for joining us. We really appreciate that you've taken the time to do that. Can I have the first of the slides, introductory slides that I'm going to tell you about? I know that many of you who are attending today are familiar with us, the La Jolla Institute for Immunology, but some of you aren't. So I'd like to give you some background. The Institute is an freestanding research organization. We have a facility, we have a building, and our purpose is to generate important new knowledge about the human immune response so we can help improve human health. That is our sole purpose. And to do that, we carry out fundamental research in model systems all the way to clinical trials, participating in clinical trials in some cases. But of course, we are not a clinic per se. So that is the space, if you will, that we occupy. We were founded in 1988. We have almost 500 employees, many with MD, Ph.D. degrees or both, or GBM degrees in some cases. And we're led by a group of principal investigators who head laboratories and who really choose their own area of research, their own passion, if you will. We also have a large and supportive governing board. We have a very good success rate of federal grants. It's not 19.6 million per faculty, it's 1.96 million per faculty, if I have the number correctly. But that's a very large number, a large amount of federal grants, which are very competitive. We're highly ranked in terms of the impact of our publications. We have a building of 140,000 square feet, if that's relevant. One member of the National Academy of Sciences and three former members of the Institute who are NAS or National Academy members, and 4 members of the the American Association for the Advancement of Science, 4 fellows elected. Our budget is shown, and you can see that the great majority of it comes from federal grants and contracts from our partners or affiliates, which include UCSD and philanthropy and others. Next, please. So we, in thinking about what immunology can do, we've organized our research into research disease centers in autoimmunity and inflammation, in infectious disease and vaccine research, and in cancer immunotherapy. And basically immunology or the immune system is important for almost every disease conceivable, because your immune cells go everywhere in the body to protect you against infection, but they also can over-respond and cause chronic inflammatory diseases, including a contribution to Alzheimer's or atherosclerosis, and as well as autoimmune diseases like type 1 diabetes. Next, please. This is our research faculty organized into the 3 research disease centers. Next, please. So when the pandemic started back in early last year, so we are 1 year into it, and there's obviously still a lot of interest, as we can tell by our audience size today. Our investigators turned their research direction to addressing SARS-CoV-2 and COVID-19 disease, and in particular, 8 investigators who have spent years developing research platforms, techniques, and expertise have moved very quickly to study COVID-19 and SARS-CoV-2. And our research has been, as Eric Zwisler mentioned, our board chair, our research has really been, had a very large impact. And perhaps nothing symbolizes this more than Tony Fauci, the longtime director of the National Institute of Allergy and Infectious Diseases, testifying in Congress and basically holding up our work. That's a paper from our lab by the Crati and Sette labs together, holding up our work and telling Congress that this work on T cells that you'll hear more about is what the kind of work that we really need to pursue. And we've received many other accolades for the groundbreaking work that's been done here on the pandemic. Next, please. So our work has impact, as you can see here, in terms of licensing agreements, patents, ongoing clinical trials, and in both in cancer, infectious disease, and other areas. It's not simply a matter of generating the knowledge. Translating it is very important and part of our mission. Next. Okay, so the main body of the presentation will be by Steve Wilson, who's a Ph.D. in immunology, came to the La Jolla Institute originally as a postdoctoral fellow. He's a long-term employee of 24 years, and he's currently our executive vice president and chief operating officer. Steve, please.
Thanks, Mitch. Let me switch over to my presentation file here. Well, thanks again, everyone. I'm going to spend the next 20 minutes or so trying to summarize the past year and what we've learned. In doing so, I'm going to really be summarizing the work of those in our infectious disease center and coalescing some of the data from outside of the Institute as well. And I'll try and point out the differences. So if we think back to a year ago, February 9th, the day, this was the front page of the New York Times. And what's kind of clear is that coronavirus, SARS-CoV-2, was thought of as an outbreak in China. And we were watching and waiting to see how that was going to evolve. And in fact, you had to go all the way back to page 13 to see more reporting on the first US citizen that had died. By the 11th, the World Health Organization gave SARS-CoV-2 a name, coronavirus disease 19, so the disease a name. And already a day later, the Jolla Institute, and first authored by Alba, who you'll hear from after my talk, had already put out a report that talked about potential targets and immune responses to this novel coronavirus. And so already we knew that this was coming, and the Institute had already begun to make an impact. Now, I'd like to frame things a little bit because while we're all feeling a bit more expert in pandemics, it's worth looking back at how pandemics have evolved and kept up with humans. In fact, if you go back to the beginning of the Common Era, there are reports of plagues or outbreaks, and you can see the death toll. And I'm gonna roll the rather constant and consistent spread of infectious disease, how it's traveled along with humans as we've transited the world. And many times they were named after a place or a particular people. In fact, with globalization, that's clearly outmoded. The other thing is, if you look back throughout the years, infectious diseases have stayed with us until they've either been eradicated or rugged herd immunity has been developed. And so what has really changed in all this is the ability for us to do something about it, to produce vaccines. And so before I get into the data and some other things, just a thought, and that is that infectious disease on the whole is going to be a constant challenge to human health. But immunity is how we'll all be able to live a long life without disease. So one more caveat before I get into things, and that is statistics. Over the past year, many of us have heard various statistics and rates and risks. And I just want to point out that there are many apples and oranges out there. One of the biggest is that previous pandemics and really large global epidemiological studies have had little or no testing while things were going on. And with the current pandemic, there is some limited testing and it's evolving. And so I'll try to frame things as I talk about what we thought, what we knew, and what we're learning in terms of these statistics involved. So early on, February 2020, a report came out, which I think summarizes quite nicely what thoughts were at the time. And that is the relative risk, in other words, the mortality rates and infectivity. And it was a comparison chart to things that many people knew. So Ebola being very lethal and quite infectious. Another coronavirus, MERS, that was in recent memory, avian flu, a serious flu, tuberculosis. And then down here at the bottom, the previous SARS, SARS-1, the new CoV-2, COVID-19, and its comparison in terms of mortality and infectivity to things like seasonal flu and pandemic flu. And at the time, we were able to estimate that COVID-19 disease was going to be more deadly and more transmissible than seasonal flu. compared to SARS-1 though, it was less deadly and less transmissible. And so the questions that were out there were, can we contain the spread completely? After all, it was in China at the time. Which people are at greatest risk and why? Can we even make a vaccine in time? And then how long will immunity last, either through infection or through vaccination? In February 2020, we really weren't talking about what the acceptable death rate would be or what the barriers we'd be willing to blow through to get back to work and do things like that. So fast forward a year, and again, when we compare some of the other diseases and disease-causing elements, indeed, seasonal flu, a bit more right in the range there, but right now in the United States, the infectivity rate is around 0.94. So just less than one person gets infected for every person that is infected. This has changed over time, but that's roughly today's rate. The mortality rate back in 2020 was estimated to be in a range of about 1 to 4%. And if we look at today worldwide, the mortality rate is about 2.2%. This fluctuates. And there's about 24% of reported cases are in current infection. But the rate here is highly dependent upon the country that you're in and the healthcare that you're able to get. The projections back in 2020 were extremely broad. It was a time that we can all remember of great concern, doubt, various convictions about, will it even head to the United States? Will it travel the world? Will we do something about it? And so the range was so broad that in fact, if depending on the source, you might see as low as a 0.1% mortality to 10% regionally, meaning that there could be less than 1 million to tens of millions of deaths as a result, estimated at the time. On the right here, what we see is a recent projection, yesterday as a matter of fact, of the rate of cumulative worldwide deaths. And the curve is starting to flatten out, and the overall long-term projections simply aren't being made. But towards the end of the talk, I'll try and wrap up with where these might go. So back then and now, we knew that the way out was your immune system, immunity. And I say that because once an infectious agent becomes global and is pandemic, you're either going to be infected or you're going to be exposed after you've been vaccinated. And so here are your two routes to getting herd immunity. You can let it go, run rampant, and it's fast. There's still going to be a significant burden economically and societally, and you're going to contend with excessive preventable deaths and sequelae. In other words, the after effects of natural infection. Whereas with vaccination, there is going to be time needed for design, testing, distribution. It's going to be expensive regardless, and there will be societal and economic burdens of disease. But what you pick up is this greatly reduced death and morbidity. Surprisingly, even today, there are questions about which is better, vaccination or infection. Well, I can tell you that as a pandemic infectious agent, if you're reading this, you're going to be exposed to SARS-CoV-2 in one form or another. And vaccination is your chance to build an immune memory without experiencing the disease, the long sequela, or death, which is not a zero probability. And importantly, long COVID and coronavirus is not a new thing. In other words, long COVID, long-lasting post-infection symptoms, even when you do recover. And this has been reported in other viral diseases in adults, including SARS, MERS, other coronaviruses, and even old reports of the 1889 pandemic that, like today's COVID-19 disease, mostly killed people who were older, and in all likelihood was a coronavirus. So I'm going to try and bottle this up quickly and say that because your risk of death from SARS is greater than zero in natural infection, get vaccinated, please. Again, long COVID, a real concern. I have the symptoms listed up here, but essentially it's this long post-viral period. And in fact, the UK study has shown that for those who have long COVID, which is in this case defined as greater than 3 months, it's twice as likely to occur in women. Over 70% of people post-hospitalized experience it. And even those who aren't hospitalized, 1.5 to 2% are there. So vaccines also cut off this long disease sequelae. So another way out, and a continuation of the theme, is to train your immune system. And here's where I'm going to pick up the pace and try and show where we come in. So if there was a game plan, you identify the pathogen and if possible, treat it while you go down the road to herd immunity. Of course, if you can, you contain it. Now, since we identified it, we can also develop vaccines, which means protection. So we can design, observe, roll it out, and get there. And it's in these areas where the Institute has played its greatest role. And it's in these areas where the coronavirus has decided to get in our way. And so the challenges are there. So what is clear is we need to get to herd immunity through vaccination so that we can control it. Eradication is probably not a likely scenario. Early on, early last year, a report came out that showed that SARS-CoV-2 indeed used the ACE2 receptor to get into cells. And so a target for neutralization, in other words, a way to get in the way of the virus's way into your cells, was found. And it was similar to the first SARS. So soon after that, LGI reports started to come along and add to the literature and add to the knowledge base and help with that design, deploy strategy for vaccination. So I already talked about Alba's first publication in February. In April, groups here showed that there are targets in SARS-CoV-2, even in unexposed individuals. This is good. It showed us that if a vaccine was developed, in all likelihood, we'd be able to mount a defense and it would be useful. This past immunity was likely due to prior coronavirus exposure. Soon after, the structure was found and the changes in structures were defined. In other words, new immunological aspects that showed us that we could target different areas and have successful vaccine designs. The Institute was marshaled to form the COVID Alliance, which is a global alliance. It put together groups, including industry and research groups who are normally competitors, to all work together in a large consortium to sort and catalog those spike footprints, in other words, the targets of neutralization, to start to look and see how various drugs and other interactions or other interventions could affect the current strain and potentially other strains, to catalog therapeutics across continents, and then put them out to groups to independently verify and compete for the best one. And then, of course, to use that knowledge to engineer even better vaccine components as we move forward. Now, by June, the Institute and other groups started to look at COVID, the disease, because there were samples that were available and patients that were coming through, to look at these various syndromes that are associated with it. And as immunologists, we were able to show how the different kinetics of disease or variation in responses may play into the pathology. And one report in June and another in July showed that different cell types in your immune system could be hyper-inflammatory and overreact. And in one study looking at not T cells, but in this case monocytes, when the inflammatory side of those cells dominated, it was characterized in moderate and severe disease, whereas another cell type patrolling monocytes were characterized in mild disease. In other words, your immune system and its healing component in monocytes, when it was favored in the response, you suffered minor disease versus moderate and severe. Soon after, in August, variants came on the scene. And in fact, the so-called G variant became the dominant strain. As you can see here, between January and April, it swept through. And we know now that that's the story of coronavirus and its natural disease evolution and change in the human population. In October, a report from LGI Labs came out and showed that blood samples from unexposed people was very similar in its ability to recognize COVID-19 or COVID-2 epitopes, in other words, targets. And so latent memory, again, was showing that the human immune system can respond to and successfully defeat SARS-CoV-2 when it's targeted. This is reinforcing data. And if we think back to that time, this is also when we were all waiting to see what were the results of the first candidate vaccines focused on these predicted epitopes, these predicted targets within coronavirus. So this was building more momentum towards what we know now, which is effective vaccines. In November, reports came out that showed why age had a role in it, and that is the so-called coordination of your immune system. When all branches required an antiviral immunity work together well, you had the best outcomes. And this seemed to be disrupted, particularly in individuals older than 65 years. And so this correlation that we saw in epidemiology was confirmed immunologically and some rationale behind it. This again allows us to build strategies to better protect those populations that are going to be more susceptible. Another report soon after showed that the imbalance of regulatory and cytotoxic, in other words, parts of your immune system that tamp down the immune system when the infection is gone, when they are out of balance, you can have runaway immunologic effects. And these runaway effects cause serious pathology and perhaps even death. January of this year now, more reports about severe COVID-19 disease and the difference. And that is, again, this lack of restraint, this inability to regulate the immune system to bring it back down in control after the virus is cleared. And this is also where we started to realize that in order to fully understand this, we cannot just study humans from afar, but we're going to need model systems that we can induce disease and then tear apart the mechanism so that we can refine things further. Certainly, we wouldn't want to infect someone who didn't already have COVID just to find out what happens. In January, additional results from the Institute were published and showed that immunologic memory, the question from early on, how long does it last? Well, so far, 8 months. And in fact, in the vast majority of people, about 6 months after infection. And so timely reports showing real data to reinforce our conviction that vaccination is going to be a long-term strategy to get us out of here. And so looking forward into the future, what do we still need to do? Well, I talked a little bit about the fact that if we're going to dissect the disease and perhaps even some of the long-term effects, we're going to need to induce the disease. And doing that in small animal models is extremely helpful. For one thing, it's fast, safe, and it doesn't put humans at risk. It will also let us take high risk, high reward angles. So imagine a universal vaccine or something that hasn't been tried in humans that we want to bring to bear. A model system will do it. And then the second is really a commitment to preparedness. And so in some ways, the Institute was prepared, but looking forward to be at the ready and to put things like our biosafety lab, level 3, in place, ready to go, so that if the next pathogen or next threat comes along and can't be even handled in regular laboratories, we have a place where we can do it. To take those lessons learned and those investments and the support that we've had and lever it so that the same discovery rapid progress that we saw this past year can be compressed and optimized further. And of course, to make sure that we've got plenty of people studying disease and not resting on our laurels thinking this is it. Simply put, being ready before the crisis. And so where do we stand now and what's going on? Well, I think the greatest amount of talk is about the variants. And so as we start to see over the horizon, what comes next? And I said that I'd get back to the projections on what was going to happen. Well, by June, the hope and the projections show that the daily deaths are going to drop off dramatically. And so through vaccination, containment, and all of the scientific knowledge and healthcare knowledge gained, that rate is going to drop off precipitously. And so we can answer some of the past questions, things like, can we contain the spread completely? No. who are at greatest risk, those older and with certain comorbidities, and we even have some ideas as to why. Can we make a vaccine in time? Thankfully, yes. Remarkably, many vaccines are coming to bear, and they're effective, and they're safe. How long will it last? Well, time will tell, but already there are good sets of data and indications that it will last quite long, a year or longer. So now the questions really centering around variants. So can we contain a faster spreading variant? Well, the race is on because it is happening. Are the same groups in these variants going to be at risk? For now, that seems to be true. We need to find out. Will we need to make new vaccines? Almost certainly yes. And as a matter of fact, the work that was done to show why these first set of vaccines were so effective will be levered so that the next set of vaccines and iterations can be done rationally, and that engineering will be a very rapid cycle where as quickly as we need to change, those changes can be implemented. And then how long will it last? Well, some of this is actually going to take the time to measure it. And so one conclusion is that this coronavirus will become an annoying childhood infection. And that's actually a pretty good outcome. In the end, if we can vaccinate most people and young kids are not infected in a very pathologically dangerous way, then yes, in fact, this coronavirus can join several others that are thought of as a nuisance, even though they're a bit more than that, but as a common cold type variant that one day will go into that catalog. So at this point, I would like to hand over to Dr. Alba Grifoni. She's an instructor and research faculty member in Alex Sette's lab and in the Center for Infectious Disease. Dr. Grifoni got her Ph.D. in 2014. She has been focused on studying viruses and viral immunity in particular. She has an informatics background. Early on after her PhD, she spent some time in industry. This is after graduating from University of Rome. She joined the Institute a few years later and is going to talk to you about her latest work and show you around the lab. So I'm going to stop here. Alba,
thank you very much, Steve.
Hey, you got it.
Mute your computer. So, hello everybody. The beauty of the live. Sorry about that. So today I'm gonna show you. There you go. Sorry about that. So welcome everybody to the Live from the Lab. I'm Alba Grifoni and I work in the lab of Cell and West Cove. And today I'm gonna show you our most recent publication that has been published actually 1 week ago, where we keep trying to understanding how immune response behave in COVID-19. And when we talk about an immune response, in reality, the immune response is a teamwork, and there are many players that are really important. So for instance, one of the components that is actually the most known is the one of the antibodies. So what the antibodies do in this case, they bind to the virus, and these prevent the virus to infect the cells. This is why the antibody response is really important, particularly in vaccination. But when the virus manages to infect the cells, the antibodies don't see it anymore. So this is where actually the T cell response play a major role. The T cells are able to recognize an infected cell and kill it. These are our killer T cells, or they're able to recognize it and understand what type of immune response is the most important to fight this specific infection. So those are the helper T cells, and those are in fact critical for the antibody response. So in our study, we've been focusing in understanding more the response of T cells. And while in the context of the antibody, the surface protein are important, when we look at the T cell response, all the protein of the virus can be virtually recognized by T cells. So not only the spike responses, but also I'm sorry about that. Not only the spike responses, but also the other type of protein. So one of the we wanted to understand is each individual, how many protein is able to recognize. And this is important because indeed all our vaccination are now composed by the spike protein, but we wanted to understand if there are other protein candidates that can be used for second generation vaccination. And we were happy to see that on average, after we studied 99 donors, 3 proteins are recognized both in the context of the helper T cells and also in the context of the killer T cells. So this is a very good news, meaning that we already know if the needs arise, what type of protein and how many proteins we can actually include in a vaccination. But the proteins are pretty big. And as we have seen previously in the context of the antibody response, not the entire protein is recognized, but just portion of it. So we wanted to understand, what about the T cells? Are they recognizing the entire protein, or do we see that there are specific regions of the protein that are important in the response? So this is a very complicated slide, but I'm gonna guide you through it, and I hope this is gonna be easy to understand. What you're seeing over here is the entire sequence of your spike protein. Here, the peaks that you see represent how strong the response of our helper T cells is. And you can visually see already that the peaks are not uniform. And indeed, there are particular regions that are very important for the T cell response, while we see other regions that are not recognized at all. So this suggests that also helper T cells have preferential recognition of part of the protein. And what is striking is that the portion that they recognize are not necessarily the same that the antibody recognize. We all know that in the context of the antibody response, the RBD domain is very important. And as you can see, we have a poor recognition from the helper T cells. So this means that when we want to design a vaccination, our decision should not be driven only by the information known about the antibody response, but we also need to take care of what the helper T cells recognize as well. And while this is particular for the helper T cells, this is not necessarily true about the killer T cells. So up to now, the insurgence of the variants are raising a lot of concerns in the community because we don't know yet how the immune response is going to behave. So while some data have been shown for the antibody response, studies are still needed for the T cell response. So today I'm going to show you how the variants are actually impacting the data that we have found up to now. And as you can see here, this is the same images that I showed you before. And now the arrows show you which portion are mutated in the context of the South African variants. And it is true that there are several arrows, but if we look more in depth, the majority of them, they actually correspond to portions where there are no high peaks, therefore suggesting that indeed the mutations are present, but they don't necessarily impact the majority of the T cell response. So we did a more comprehensive analysis looking not only at the South African variant, but all of the variants currently concerning. And we were happy to see that 90% or more of the findings that we found are still conserved, therefore suggesting that we should still have a good T cell response even when an individual encounter COVID-19 variant. Of course, these are hypotheses, and we are working to actually perform experiments to prove this point. So stay tuned, and maybe on the next webinar, we're gonna be able to tell you more. So when we work on the T cells, there are actually different types of technique that we can utilize. And today, I'm gonna show you the fluorospot technique. So here is our fluorospot machine. And what we do, we use this type of plates and we add the cells of our individuals and the peptides corresponding to the proteins. Now, if we put our plate inside, the fluorospot instrument is gonna able to measure our T cell response. So here is the results of one experiment. And as you can see here in green, what you see is the production of interferon gamma that is a critical cytokine for the T cell response and is correlated with protection. So here we test the spike protein. As you can see, with very strong and beautiful response. But how do we understand that this is a significant response? So first of all, we repeat our results 3 times. So we want to make sure of what we see. And second, we use 2 types of control to make sure that this response is indeed effective. One is called the negative control. So we are adding our cells, but we're not adding the spike protein. And see, we see no green spots at all, meaning that indeed the response we saw before is real. And the second type of control that we use is a positive control. So we want to make sure that our cells are able to respond to a stimuli that should be able to let the T cell respond normally. And there you go. This is our positive control. And beautifully enough, is as strong as the spike protein. So we really have a very nice T cell response against the protein. And with that one, I give the speech again to our president, Mitch Kronenberg, and thank you very much for joining us today.
Hello, everyone, again. Thank you, Steve, for a great overview of what's been done at the Institute for the SARS-CoV-2 COVID-19 disease and the research that we are doing and how it's had an impact and how we might look at the future. And Alba, Dr. Grifoni, thank you very much for your presentation and for taking us into the lab. Could I have a slide, please? Yeah, thank you. So we are, as I mentioned, we are a freestanding institute. In other words, with our own governance, our own board, and our board chair, Eric, who started us off today. Thank you, Eric. Our own finances and a building that we occupy along with one of our partners. And we're not very big. So how is it that we've been able to have such an impact on research onto this epidemic and gotten attention from Wall Street Journal and New York Times and or whatever your favorite alternative media might be? And there are really 2 reasons. One is our investigators have spent years building up expertise in figuring out how to analyze the responses of the immune system, how to identify the peptides that are recognized, as Alba showed you, how to identify what antibodies recognize, how the B and T and other cells of the immune system respond to virus. Having this knowledge accumulated over years has been critical. And the other thing that has allowed us to start so quickly and to be so effective is philanthropy, is private giving. We are mostly funded by a competitive federal granting system. And as I mentioned in the beginning, we are successful at doing that. But that system takes time to roll out and to receive, review, and fund grants. The government in Washington moves at its own speed. Our work has been accelerated by philanthropic donations, essential in a time of pandemic. These donations have allowed us to buy critical instrumentation, to build out our biosafety level 3 facility where we grow virus. And it's now, donations are now allowing us to expand our clinical center so we can process blood more efficiently from infected people or recovering people from the pandemic, as well as others. But there are always needs at an organization like ours, a nonprofit organization. And right now we're in the midst of recruiting Sam Myers from the Broad Institute, which is a joint effort of Harvard and MIT in Cambridge, Massachusetts. And Sam is bringing to us a mass spectrometry technology that allows us to understand how proteins interact and signal to one another in detail. How proteins of SARS-CoV-2, for example, interact with other proteins in the blood or on cell surfaces. Sam's recruitment is a cost of over $3 million to the La Jolla Institute for Immunology. Sam will get grants. He's a new investigator, a young postdoc, just finishing his training, a fresh mind to really augment the resilience of the La Jolla Institute for Immunology. but he doesn't have grants yet, and his work needs to be supported. We've raised the money for the mass spectrometer, so the 1.7 million, but we haven't raised all the costs for bringing Sam here and running his lab with an anticipated arrival of March 31st this year, so coming right up. So if you are thinking about philanthropic gifts and you are thinking about responding to the pandemic generously, I hope you'll consider the La Jolla Institute and consider helping us to complete the funding for the recruitment of Sam. And now I think the next step will be for us to have a moderated question and answer session, which will be led by our board chair, Eric Zwisler. Thank you.
Thanks, Mitch. I really appreciate it. So we have Steve Wilson. Mitch, you're going to stay on, and Alba, you're going to stay on to answer questions. I hope I'll toss them out there and hope you guys can go for the jump ball if it's in your area. Okay, I'll try to direct them. We have a lot of questions, and so we've got probably about 30 questions here. So I'll try to combine where I can and try to answer some. If you guys can keep your answers relatively to the point, that would be great. So here we go. And we have we have about 10-15 minutes to run through this, and hopefully we get some insights. I mean, thanks to the SARS-CoV virus, it's a gift that keeps on giving. So we're never at a loss for things that we want to understand. So real quick, lightning round here for the three of you. Is there any reason not to be vaccinated by any one of the vaccines? Are there any safety concerns, particularly questions about the mRNA new technology? Mitch, you want to handle that?
Well, there are probably a few exceptions. There are some lipid components that envelope the RNA, and there are people who are known to be allergic to those components, which is why they have you, even if you're not known to be allergic, they have you wait 15 minutes to make sure you don't get a severe allergic reaction called anaphylaxis. If you're known to have such an allergy, you should avoid Pfizer and Moderna vaccines. This is very rare, but it happens. I think there's a little bit of uncertainty about pregnant women or very young children, but vaccines will be probably moving into those populations. Otherwise, perhaps some severe immune deficiencies. You won't be at danger from an RNA vaccine, but we can't anticipate you'll make a response and benefit. Those are a few of the exceptions. There aren't many.
Would you have, Steve, Alba, would you have any concerns about taking the vaccine yourself? Any preferences?
I'll answer. So Eric, no. So vaccines, as Mitch said, there are a few groups who either are allergic or have other issues, but pretty remarkably and well documented, these are not more dangerous than other vaccines, which are extraordinarily safe. In other words, what we're talking about is the relative risk, right? The risk of not getting vaccinated and winging it with disease, or getting the vaccine. And in this case, it's absolutely clear that being vaccinated is better than taking your chances with natural infection.
Okay, good. Can you comment, Alba or the other two, Steve or Mitch, on after vaccination, on whether you can still transmit the virus?
Yes, I'm happy to take that question. So up to now, the majority of the information that we are aware of is that these vaccinations are able to protect us from disease severity. We don't know yet whether the vaccination is going to be able also to protect us to get asymptomatic infection. Therefore, when you get vaccinated, you're not going to develop disease severity. However, you can still transmit the virus to the people around you, which is why it's important to keep wearing the mask and practicing social distancing.
Okay, great. Is it safe? Here's another question for you on vaccines. Is it safe for pregnant women to be vaccinated? Steve, do you have a comment on that?
Yeah, so right now, there hasn't been exhaustive studies, but in all likelihood, it will be safe. As a matter of fact, maternal antibodies might become quite helpful to the developing fetus and early childhood, as it is with other vaccines in pregnancy. But those specific studies are not out yet. There hasn't been any reports, though, that are significant enough to create any warnings, which by now you would expect those to be out there. Pregnancy is being tracked and vaccination status.
Yeah, pregnancy is an exclusion criteria, but the populations tested in the trials were so large that there are people who didn't know they were pregnant at the time that turned out to be pregnant. And we don't have reports of adverse effects. But there, as Steve mentioned, we haven't done a controlled, no one has done a controlled trial in pregnant women, and we want to be especially careful in that group.
Okay, good. Alba, can you comment at all on the length of immunity? We have a number of questions here about the length of immunity, either post-infection from COVID-19 or whatever we know on the various vaccines. In other words, what length of immunity can we expect, or do we know will be there?
So recently our lab and Shinkrati lab has been actually trying to address this question in the natural infection. And what we see is that 8 months post-infection, we are still able to detect a protective immune response, therefore suggesting that up to 8 months, we do see that. Of course, the virus is not circulating from enough time to go more than 8 months, but we'll keep tracking these donors to understand how the length is gonna be. As per the vaccination, we might hypothesize that it should be able to mimic the same length of protection of an immune response, but it's early to say how long it's gonna last for the vaccination as well. So we will need more studies to address this question more carefully.
Okay, good. Just a comment here. We hear a lot about vaccines and about the efficacy of vaccines at 70%, 90%, 95%. How does that translate? I mean, can you just briefly explain, whether that effectiveness at 60 or 70 is still important and relevant, and what does it actually mean? So in other words, if you're 95% effective, what portion of the vaccinated population is it ineffective for? Alba, can you handle that?
Oh, yeah, of course. So the type of study that have been done, they follow people that got the vaccination and people that didn't, and they wanted to see the insurgence of disease severity. So when we talk about an effectiveness of 95%, means that 95% of the people that received vaccination did not develop the severe disease. So of course, the type of study varies a lot depending on the type of clinical trial and the endpoint that has been done up to now. But this is what we meant when we talk about efficacy. There is still a small percentage of population that even with vaccination might lead to the disease severity, but we are decrementing considerably this percentage with respect to not getting a vaccination.
Okay. Steve, there's a question here on the RO rate of COVID-19 virus. Somebody's been going through the historical record and said about a year ago, we thought it was probably somewhere between 2 and 2.5. And the data you put up generated was, I think was a bit less. Can you just go over the development of what we've learned and where that infection index is today?
Yeah, so the rate that I showed you is the actual rate that's being measured in official reports. In other words, when you compare the number of people known to be infected versus the next wave and on, it changes. And that rate is actually regional. And so in different parts of the world and different parts of the United States, the rate is different. And so the infectivity of the natural virus, where you would just allow people to run wild and infect, is yet to be determined. It's probably higher than 1. As a matter of fact, if you put someone in a room with 5 people long enough, they'd all get it, and then you'd have an R of 5. But the rate that I was showing up there is actually the rate that we should all think about. In other words, in real world conditions and under what we're living now, a pandemic, what's the growth in infection versus the contraction in infection? And over time, we're going to find out what that natural rate is everywhere in the world, and then come up with it. So again, apples and oranges. I think what's key here is that it's held up that it is quite transmissible, but not in the likes of measles or some of the others that, you know, hours and even a day after someone leaves the room, they can be infected. So it's nothing to sneeze at.
Yeah. Can Mitch, can you comment on vaccination and autoimmune diseases, and then I'll follow up with another question here. We had a specific question about COVID-19 cytokine storm sequelae increasing the incidence of type 1 diabetes without evidence of typical autoantibody precursors. Is it possible that this kind of type 1 diabetes may be reversible? And so first, autoimmune diseases and vaccination. And then specifically, have you heard or understand anything about the cytokine generated type 1 diabetes?
Yes, thanks, Eric. I don't know of any autoimmune disease where vaccination would not be recommended. The vaccine directs your response to a very limited set of determinants, molecular structures, proteins, whatever you want to call it. And by itself, it shouldn't greatly exacerbate autoimmunity. And I don't know of many examples where that's happened. And I don't know of any recommendation where if you have this or that autoimmune disease, you should not be vaccinated. So that would be my answer to the first part. Regarding the cytokine storm and transient autoimmunity, it is possible that that type of autoimmunity that's induced in rare cases is more reversible. We've seen that in some of the cancer immune therapies, which basically unleash the immune system to fight cancer, and you do get autoimmune sequelae, and those have turned out to be more treatable than the normal course of autoimmune disease that develops over decades. But I think it's too early to be certain about that. And as I want to emphasize, as far as I know, the cases in which that has occurred are fairly limited.
Okay. Any updates from the three of you on hydroxychloroquine and COVID-19? You know, we were up, we were down, clinical studies had difficulties. Could you comment at all whether there's any new learning or whether we should change anything we understand now? Steve, are you able to answer that?
Well, I can talk about it a little bit, but I'd rather not make any definitive statements, only because it's an ionophore, and if you can precisely get zinc in there to interrupt viral replication, that's a good thing. I think that the confounding element of it is there have been studies, there has been controlled work. It's been looked at in detail to see if, you know, treatment with hydroxychloroquine upfront is going to protect you from viral disease. And the answer, unfortunately, was no. And because of that, it's at this point not reasonable to think that that is a substitute for something else. And it's not to say that in the future it won't be an adjunctive therapy or it won't be something that's part of treatment. But those studies are gonna have to really clarify how to do it. Yeah.
Let's try to do two more questions here. I know we're going a little bit over time. We should just do a live from the lab, which is Q&A, which would, because we have so many questions here. Steve, you mentioned in your talk that you hope that the COVID-19 would become a childhood troublesome or irritating childhood disease. Can you just expand on that a little bit, and then at the same time talk about you know, the relationship with controlling this disease in the future and booster vaccinations?
Sure. Yeah, so in brief, as the world becomes immune to the current virus, the virus is going to try and survive. And in doing so, its most likely path is going to be a non-lethal form or a less lethal form that still allows it to replicate and transmit and be out there. And really what the strategy here is rather than a massive die-off, in other words, we let the virus run through and kill everybody who's not resistant or can't fight it, we protect them. And then, as we've seen in other coronaviruses, young children don't have the same pathologies from disease. They become immune through exposure or perhaps childhood vaccines, and they're immune. And now, as we fast forward in the future, the world is full of people with immunity to COVID-19, to SARS-CoV-2. And so then, as each seasonal wave comes through, we become infected, but our immune systems are ready to go and we don't feel the effects of it, or it's a nuisance and we're over it. And so that's a hopeful outcome. The thing that we need to stick to though, Eric, is it's novel right now, which means right now, not everyone is going to survive. Not everyone is going to have a good outcome. And so we have a duty to prepare and protect those folks so that we don't see millions of deaths that can be prevented.
Okay, great. Great. And our last question, I mean, the flavor of the day that we've all been talking about has been vaccines. Would any of you care to talk about therapeutics? And is the future really going to be around vaccines, therapeutics, or the easy answer is probably both, right? But could you please comment?
If I could start, I think therapeutics are a really important area. There are people who may not respond well to the vaccine if it's 1 in 20, and perhaps the 1 in 20 that didn't respond have reduced symptoms, but still people of a very advanced age. The vaccines seem to work well in people over 65 and over 75, but perhaps in older people and others. Therapies will always be important. They could be monoclonal antibodies. They could be antiviral drugs like remdesivir. It could even be something like dexamethasone if somebody gets into this hyperinflammatory state that the very sick people get into. And then there are other things that people are speculating about. We talked about hydroxychloroquine, which hasn't worked out in most of the tests that have been done. A lot of these things haven't been through advanced trials. Fluvoxamine, which is for obsessive compulsive disorder. There's some very early stage work on 6 patients. Ivermectin, some people, which is an antifungal drug. These things really haven't been through the rigorous testing that the vaccine has undergone. So I think for therapies will improve. They will be part of the armamentarium that will allow us to fight this. But ultimately, the most cost-effective way to fight this is not to have the person in the hospital getting dexamethasone or remdesivir, but to be able to protect the people through vaccination and herd immunity.
Okay, fantastic. And I said it was the last one, but I'm going to ask one more question here. And maybe Alba, we haven't heard from you. I hope this is up your line. What about vaccination of children? Can you comment on safety and effectiveness, please?
So there are studies ongoing to actually address this point. So there are no indication for now that it's not going to be safe, but it is important to wait the results of these studies before reaching some specific conclusion. It was a short answer.
No, it's a good answer. Short answers are good. But anyway, listen, I mean, I always learn a tremendous amount and I get to talk with everybody there really often. And so I really do appreciate it. Alba, thank you particularly for allowing us into the bowels of the research beast. I mean, to see what actually goes on to generate those black and white articles and the talking heads we have on TV. without your hard work, I mean, we wouldn't be anywhere. And I will say that it's not as if the La Jolla Institute started in January last year to research these areas. A lot of what's happening right now is the confluence of ongoing research that has taken years and decades to do. And it's people like Alba that have been leading that effort and the charge to really have these insights, make a vaccine available. I want to thank you all for the questions that you've provided. We've had probably a little over 50 or 60 questions. I've tried to group some of them. Those questions that weren't answered, we will endeavor to get back to you directly with answers to those. We really appreciate your engagement on this. Mitch, Steve, I really do appreciate your leadership of the Institute and taking us to this point and the contributions that everybody there has made. It's a complex endeavor. Please note that this presentation, for those of you who have registered, you'll be getting an email and context so that you can watch it again and slowly read the slides that we had. And that'll be emailed to you. It will also be available on our website, www.lji.org. And if you're so moved to really help us on this journey and continue to answer these questions in the future, we surely would like your support, large and small, in any way you can to advance, to continue to advance, our journey on public health. With that, I'd like to thank you all for attending. I'd like to thank our panelists, Alba, Mitch, Steve, and everybody behind this, Chris, and his whole team. So I wish you a good afternoon and be safe, be well, and hopefully we'll get through this all together. Goodbye.
Thank you.
Thank you. Bye.
Yeah.